So today our topic is electrophoresis. It is one of the important topic when we are dealing with the charged molecules separation. So in electrophoresis, the separation of charged molecule take place. Separation of charged molecule. Charged molecule means that when electricity is applied, then the molecules separate. So in electrophoresis, what happens is the relative mobility of individual molecules. It depends on several factors. Individual mobility or movement of individual molecules. When we are dealing with the individual molecules, so there are certain factors which are responsible, and these factors are net charge, charge mass ratio, molecular shape, temperature, porosity, and viscosity of matrix. So these are certain factors which are responsible when we are providing electricity and the separation of molecules are taking place. So many important biomolecules are there, such as amino acids. These are the biomolecules such as amino acids, proteins, peptides, nucleic acid, are separated by electrophoresis. These molecules are separated by electrophoresis method. And why they are separated by this method? Because these all molecules, amino acids, protein, peptide, nucleic acid, nucleotides, these all molecules carry charge. So what we have read in our first definition is that the separation of charged molecules, like when the molecules are charged, they are separated. So these molecules, amino acid, protein, peptide, nucleic acid, they all carry charge, and when the electricity is applied, they all separate. These molecules either migrate to cathode or anode, either migrate towards cathode or anode. Cathode carrying the positive charge, cathode carrying the negative, and anode carrying the positive charge. So this phenomena, which means that these molecules, the above molecules, amino acid, protein, peptide, nucleic acids, are separated, and they either migrate towards the cathode end or the anode end. This is called as electrophoretic mobility. When we break the term electrophoretic mobility, which means the movement of molecules when they are applied, or when the electricity is applied to it, they move towards either cathode or anode depending on the charge which they carry. So now we are going to discuss about the types of electrophoresis. There are many important types of electrophoresis which we are going to read and learn in detail. So the first one which we are going to read is age. This is one of the most important type of electrophoresis. It stands for Agarose Gel Electrophoresis. It stands for Agarose Gel Electrophoresis, and this type of electrophoresis is used for the separation of DNA and RNA, which are our nucleic acid. So this age method of electrophoresis is applicable for the nucleic acid separation. So what happens in age is that this gel electrophoresis of nucleic acid is based on charge and size. So this method is based on charge and size of nucleic acid which we are studying, either be DNA or RNA. So we are going to take the example of DNA. So DNA, it carries the charge. As we all know that DNA carries charge, negative charge it carries, fine, and it is separated on the basis of their length. DNA is separated on the basis of length. Separated on the basis of length. So now we are going to make a setup with which you can understand clearly the process of agarose gel electrophoresis. So we are going to create a setup. Suppose this is a buffer medium. Buffer is used to, or its function is to maintain the pH. So this is our buffer medium which is filled inside this setup. Now what we are going to do is we'll place agarose gel. This is our agarose gel system. Fine, this is our agarose gel. This is our agarose gel, and now what we'll do is that with the help of comb, this is a comb like structure. We use a comb in order to make grooves into the agarose gel. This is a comb like structure and which is used to create grooves in the agarose gel. These are the grooves. So the first groove, over here we place the known DNA. Sorry, let me take another color. Over here we take known DNA sequence, known length of DNA sequence we take in our first groove. Alright, and over here in this and this unknown sequence which we have to study, we place DNA of that sequence in these two groups. So this is a known sequence, the sequence which we actually know. This is our negative, and this is a positive terminal. As we all know that the DNA is negative charged, so it will migrate from negative to positive end, from cathode to anode. Now in the setup itself, we know the sequence of the first groove in which we have placed our known length of DNA sequence. So 100 base pair going up to like 500 base pair. So in the second one, in this one, when it will separate like the larger molecules of DNA. Suppose there is a DNA and it has a larger molecules of DNA, it can easily pass through the gel pore. So in agarose gel, it has pores. It has pores. So the DNA, the larger DNA molecule, like the larger DNA or the more appropriate term would be longer DNA molecule cannot easily pass through the gel pore because of its length, the agarose gel pore. Whereas the shorter DNA molecule can easily pass through the pore. And with the help of their differential sizes, suppose over here the larger DNA molecule is present at the top, it is unable to move downwards, whereas the smaller DNA molecule over here, suppose the smaller DNA molecule can pass to the bottom, like it can pass further, whereas the large DNA molecule cannot pass. Same case with the third groove in which we have placed the DNA molecules, unknown DNA molecules. So what we can do is that by their differential sizes, as you can see that the size are different, so by the differential sizes or the difference in the sizes, we can locate the position of DNA in agarose gel matrix. So over here it is the entire setup, and next thing is that the concentration of agarose can be varied. You can change the concentration of the agarose gel if you are taking the higher concentration. Suppose you are taking higher concentration of agarose gel, then you will get a smaller fine pores. But if you are taking the lesser concentration of agarose gel, then you will get larger pore size. It all depends upon what kind of DNA and like from what species you have isolated the DNA and you are studying. It completely depends on that. And the next question which arises is that why do we not use water instead of buffer? Why not water instead of buffer? I think that you guys might have this question that why we don't use water instead of buffer. The simple answer to this question is that buffer, it is a salt solution we are taking, and the salt solution, it contains electrolytes. This salt solution contains electrolytes, which is important for conductance, for bringing DNA from negative to negative to positive end. Whereas water, it doesn't work like that because it does not have the salt solution. So that's the reason why we are not taking water and we are preferring buffer. Next is how agarose gel is prepared. Agarose gel preparation. Agarose gel preparation. What is agarose? First of all, the question is that what is agarose? It is a polysaccharide. It is a polysaccharide and it is extracted from seaweed. It is extracted from seaweed. It is a linear polymer. It is a linear polymer made up of repeating. It is a linear polymer made up of repeating agarose unit. And this agarose unit is a disaccharide. This agarose unit is a disaccharide which is made up of galactose and 3,6 anhydrogalactose. So dry agarose suspended in buffer solution and it is boiled until we get a clear solution. So it is a process in which what we do is generally in like electrophoresis, what we do is we take 1 to 3% concentration of agarose we use. Second, what we do is dry agarose. We take agarose in a dry form and it is suspended. It is left in buffer solution. The solution which is a salt solution. We leave our dry agarose in the buffer solution and we boil it until we get a clear solution. Now what we do is, after suspending the dry agarose in buffer solution and boiling it until we get a clear solution, and after that what we do is we allow it to cool. So now we'll get our agarose gel on which process of electrophoresis can be carried out. And now buffer solution, as we are talking more and more about buffer solution, so we need to know that what buffer solution contains. We know that it is a salt solution, but besides salt solution it contains many other things also. So buffer solution, which is a salt solution, it contains sucrose, glycerol, ficol, bromophenol blue, which is a dye, and the gel is stained with ethidium bromide. Now we are talking about the gel, the agarose gel. It is stained with ethidium bromide, which is an intercalating dye. Fluorescent dye, it gives a color when we observe our DNA bands under the UV rays. So when agarose gel, it contains many things it is containing ethidium bromide, which is an intercalating fluorescent dye. So this fluorescent dye, this particular fluorescent dye, this intercalating ethidium bromide, which is a fluorescent dye, it is the dye which is showing the color, it is showing the trans illumination, because of which we can determine our DNA or the RNA bands. So it is about the agarose gel, which one of the type of electrophoresis. So our next topic would be PAGE, that is polyacrylamide gel electrophoresis. So once more, or the revision. Electrophoresis, it is a separation of charged molecules. When electricity is applied, the individual molecules separate on the basis of the charge they are carrying. So there are certain factors on which the mobility depends. First is net charge, second charge mass ratio, molecular shape, temperature, porosity, viscosity of matrix. So what are the biomolecules which can be separated by electrophoresis? These are amino acids, proteins, peptides, nucleic acid, nucleotides are separated by electrophoresis. These all molecules carry charge. These molecules either migrate towards cathode or anode. Cathode, which is negatively charged, anode, which is positively charged. This is known as electrophoretic mobility. Types of electrophoresis. So we have discussed one type, that is Agarose gel electrophoresis, and this electrophoresis is applicable for the DNA and RNA separation and identification. Charge and size of nucleic acid. So the separation is based on the charge and size of the nucleic acid. DNA is negatively charged and it is separated on the basis of the length. So it is the entire setup which we have prepared for the separation of the DNA molecule. So we have used a buffer which is important for maintaining the pH. We have used the positive and negative cathode and anode. We have used the agarose gel on which the entire process is taking place. Now the agarose gel, it has pores. The agarose gel, as you can see in the setup, it is having the pores, and you can see the larger molecules cannot easily pass through the agarose gel pores, so they remain behind, whereas the smaller pores, the smaller DNA, it passes through the smaller pores which are present on the agarose gel. So by comparing this and by the differential separation, we can see the concentration, or we can do our work. Why we cannot use water instead of buffer? Because water, it does not have salt solution, whereas buffer, it is a salt solution carrying electrolyte. Agarose gel preparation, it is a polysaccharide derived from seaweed. It is a linear polymer which is made up of repeating agarose unit, which is a disaccharide and which is made up of galactose and 3,6 anhydrogalactose. We use 1 to 3% concentration of agarose, and the dry agarose is suspended in the buffer solution, and it is boiled until we get a clear solution, and it is allowed to cool. Now agarose gel on which the process of electrophoresis can be carried out. Buffer solution, it has sucrose, glycerol, ficol, bromophenol, blue dye, agarose gel, ethidium bromide, which is an intercalating fluorescent dye, is used, and it gives a luminescence by which we can determine the DNA sequence and the DNA identification we can do. So this is about the electrophoresis. I hope that you've understood the concept of electrophoresis and one of its type, that is agarose gel electrophoresis. If you have any doubt related to this particular topic, do comment and I'll try my best to solve it in a limited span of time. Thank you.